Hi students, hope you are doing well. Welcome to Turito live classes. Myself A. Swagat, I am the one who will be discussing the biological topics with you. So in this particular session, we will be having an understanding about some of the cellular organelles, and before going to those topics, we will be having a detailed understanding of the structure of cell, and we will try to compare the striking features of plant cell as well as an animal cell. So like I said, you can have a look at the topic given here. So we are going to look at those topics and we will be proceeding the session in that way. Firstly, we will try to look at what we are going to learn now. As I said, what we are going to learn now, I am going to show you the learning objectives for this particular session. So we will be exploring all the organelles of the plant cell and an animal cell. Once done, we will try to compare the striking features of the plant cell as well as an animal cell, and later we will try to start analyze the detailed understanding of different organelles. So the later sessions will also be followed by the understanding of several organelles. Today in this particular session, we will be having an understanding of organelle mitochondria, endoplasmic reticulum and ribosomes. So this is what will be our understanding for today. So before going to these details, we will be looking at the topics what we have already covered so far. So in the previous session, we had a detailed understanding of cell wall, which was an additional membrane in the case of plant cells. In the same way, we find the cell wall, bacterial cells and fungi cells or fungal cells. And we also understood the significance why there is a need for the plant's bacterial cells and fungal cells to have an additional membrane. I mean, in order to the plasma membrane, in addition to the plasma membrane, the cell wall is surrounding the plasma membrane. Why is it required? So that is what we understood. Later, we try to understand different layers of the plasma membrane, different layers of the cell wall, where we started with middle lamella, primary cell wall and secondary cell wall. Once done, we have understood in the previous sessions that the plasma membrane and the cell wall, in the case of plant cell, they both are used to confine a dense fluid like substance inside the cell known as protoplasm, and that protoplasm is the living component of the cell. That is what we understood. And in this particular session, in the previously, we try to understand the very same point, and then we try to analyze it by separating the nucleus out of it, and we understood some part of the cytoplasm. So we had a detailed understanding of some of the cellular components, starting with cytoplasm and the nucleus. So hope you remember these topics what we have already discussed in the previous session. Now here today, like I said, we will be having an understanding of the plant cell and an animal cell, then we will move forward to compare their features what they have. So I would like to begin my class with this particular image. So hope you can see this clearly and you understand that as it is written here. This structure what you are looking here is actually the animal cell, and here one by one we will try to look at its features along with some of the organelles given here. So if you observe here, starting with cytoplasm, which is definitely not new to you. So we learnt it. So this is one such fluid which is actually, it suspends all the organelles in it. In other words, all the cytoplasm in that would be suspending, all the organelles in the cytoplasm would be suspended in that. Same stands for nucleus, and inside the nucleus you would find the presence of nucleolus. So not necessary, like in this particular image, you may find only one nucleolus. It is not the case. You will find more than one nucleoli in the case of nucleus belonging to either a plant cell or an animal cell or a fungi cell. So as you all know, nucleoli or nucleolus are actually the factories of ribosomes. So those ribosomes are made in there, and you can see here they are shown as the ribosome as a small dot like appearance. Now, the very same dot like appearance you must see on the surface of this particular structure, and that is our rough endoplasmic reticulum. Rough endoplasmic reticulum, shortly known as RER. So, that is what it is known as shortly, and there are other parts which are having no ribosomes on the endoplasmic reticulum, known as smooth endoplasmic reticulum, shortly known as SER. Once done, if you see there are other organelles, that is centrioles, combined together known as centrosome. So, a centrosome contains a pair of centrioles, that means 1 plus 1, there are 2 centrioles in one centrosome. Then we have another system here known as Golgi apparatus. As you can see, it secretes some of the vesicles and it receives the vesicles also. So, it is a system which actually designed to transport the substances within the cell, pack the substances, store it, send it to the destination within the cell, and if required outside the cell. So, it sends the substances outside the cell also. Now, once done, you see that there is a presence of peroxisome, and you see the powerhouse, which will be our topic of discussion. So, hope you know what is powerhouse. Do you know what is a powerhouse in the cell? Okay, I see some of you are responding for that. So, like you said rightly, it is the mitochondrion, because that is only one in count here. So, it is mentioned as mitochondrion. So, commonly if there are, you may not expect only one in a single cell, you will certainly expect more than one in a single cell. So, you may find it as mitochondria, okay, that is a plural. So, this is a structure of the animal cell, and all of these components of the animal cell, including the cytoplasm, are confined by a single membrane, and we had an understanding of that membrane previously, that is cell membrane. In other words, that is known as plasma membrane. Okay. So, that is what we understood in the previous session. Now, let us look at, as a contrast to this particular structure, we have to have a detailed understanding of this one too. Now, this is the plant cell. We are going to look at it and understand some of its parts. So, here are we. So, you might, let us start with the main difference what we can spot whenever we look at a plant cell, that is the cell wall. So, this part is its cell wall, okay, and you will find this being the adjacent cell wall. That means there is one cell wall, as linked to it there is another cell wall attached to it, which is actually showing the cell wall attached to the cell of this particular one. That adjacent cell wall belongs to another cell. Now, there are small dots over here, pale green in color, you can see. Actually, they are not dots, they are actually openings, they are direct openings. So, previously we learnt about these openings, and they are known as plasmodesmata, it is mentioned here. So, now you might have come across the real understanding of plasmodesmata, because previously we understood that with this particular structure, if you remember, there were two cells drawn and the structure was shown like this, and into which we have seen that some of the substances, they were kind of passing like this. So, if you remember, this is what I shown you as plasmodesmata. Now, if you look here, these encircled red colored ones here are actually the plasmodesmata, and that is how they are designed. Okay. Now, so you see that is the cell wall, following which internally there is a yellow layer over here, you can see that. That is our cell membrane or plasma membrane. So, that is what it is present, and you can observe there are some of the common organelles like ribosome, endoplasmic reticulum, which is rough in color. We saw the nucleus in the previous animal cell, and here also we can see the same, and the understanding about nucleus stands same in this particular cell also. which means you will find more than one nucleolus in a nucleus of a plant cell also. Okay. So, you see the nucleus having the pores on its surface, and that is what is nuclear membrane, which is confining this chromatin. That is nothing but the thread like arrangement we have seen inside the nucleus, that was done in the previous session actually. Now, we do have the smooth endoplasmic reticulum, cytoplasm, of course the common thing. One contrasting thing what you can find here when compared to animal cell is central vacuole. The vacuole what you are seeing here is quite large, it is quite large in size, okay, and you can see, okay, I am just erasing these things because they might confuse you. So, that is the reason why I am removing them. Okay. So, central vacuole, you see, that is being the contrasting component here which is present. This is what you have not seen in the case of an animal cell. You did not find vacuole there. So, we will try to understand why we did not see vacuole there and why we are having a big vacuole over here. Okay. Once done, I want to show you Golgi apparatus. This particular structure is the Golgi apparatus, and of course the same mitochondria what you have seen in the animal cell and even here. So, remember children, whether it is an animal cell, a plant cell, bacteria or fungi, you will find the mitochondria being at its own position, and that is one which actually provides the energy for running out all the cellular activities. Now, if the cell has to carry out its activities, it needs energy, and that energy is produced by this organelles, that is mitochondria. So, this is what some of the features of animal cell and the plant cell we have seen, now followed by which we will really look at this particular flow chart, I am sorry, the table, where we will understand their differences. So, in the case of plant cell, okay, we will find the presence of a contrasting feature again which I did not notice in the previous structure, that is the presence of the plastid. There are different types of plastids available. Okay. So, we will be having a detailed understanding of plastids in the upcoming session. So, here you see one of the plastid what I am mentioning is about chloroplast, which certainly you will not find in the case of animal cells. So, they do not have plastids. So, in the case of plants, we have a cell wall like we discussed, and plant cell wall is composed of cellulose. In the case of animal cells, certainly you will not find, okay. So, you do not have a cell wall in the case of animal cell, and we saw this plant cell having a large central vacuole. In the case of animal cells, usually you will not find vacuoles, but even if you find them, they will be available for a quite limited period. That is why there it is mentioned as temporary in nature. So, that is quite temporary in nature. So, this is what I am speaking about. Now, so may have plasmodesmata. So, we have seen the presence of plasmodesmata, but here it is mentioned as may have, in the sense you may not find all the cells of the plants must have plasmodesmata. Only the regions where it is highly required, that is where you will find the presence of plasmodesmata, and that direct connections between cell to cell is absent in the case of animal cells. In animal cells you will not find the presence of the cell wall. So, if you remember the previous class, we learnt about the cell wall in detail, and we understood it is actually the middle lamella giving rise to thread like substances and forming this plasmodesmata. Since you do not have cell wall in animal cells, certainly it is quite obvious that we will not find plasmodesmata in animal cells, right. So, next you do not have centrioles here. So, that is quite contrasting. Here we have a pair of centrioles within the centrosome. So, that is what I have shown you. I told you when we were trying to explore the animal structure, animal cell structure, I told you that there exist the centrioles inside the centrosome. There are two centrioles in one centrosome. That is what I said. Now, that is what it is mentioned here. They have a paired centrioles within the centrosome. Okay. And here you will not find the cholesterol in the cell membrane. It is just saying that here you will not find the cholesterol in the cell membrane of the plant cell, you know. So, that is fine. It is just mentioning about the cholesterol. Okay. So, you want to understand something here, that cholesterol is present in the case of animal cells. So, you find the cell membrane of animal cells having cholesterol. Now, this cholesterol, lipids, you know, they all are, you know, oils, they are components of you know, the oil what we consume, okay, that greasy liquid. So, that is of different types. So, here you will not find that cholesterol, and there you will find the presence of cholesterol in the cell membrane for the animal cells. Following which, here it stores excess glucose in the form of starch. So, plant cells store excess glucose in the form of starch. Now, what does it implies? It implies that starch is the reserved food material of animal cell, sorry, the plant cell. So, starch is what we find in rice grains, wheat, maize, corn, bajra, all of them. They are actually the reserved food material of plant cells. When it comes to animal cells, we actually store a whole lot of glycogen. So, in your body, in my body, whichever animal you think of, it actually stores excess of glucose in the form of glycogen. That is its reserved form of energy. And here you have, generally the plant cells have a fixed shape. Mostly the shape would be regular, but in the case of animals, let me tell you, you will not find a regular shape. The shape would be amorphous. That is also having a specific reason, because in the case of animal cell, you will not find the presence of cell wall, thereby making the cell is so smooth. It is not rigid like the case with the plant cell. That is one such reason why you might find a fixed shape in the case of plant cell, which is absent in the case of an animal cell. So, hope you understood the differences and some similarities what we have observed using the structures and by using this particular table. So, let us move forward and have a detailed understanding now. So, here you see, like I mentioned, we are going to have a look at one of the organelle, that is mitochondria. So, it is a singular word. If there is only one mitochondrion, you would call it as mitochondrion, or else you would use mitochondria being the plural addressing. So, Colliker was the first guy to observe this particular organelle in 1857, okay, and later there was another man called Richard Altman. He named them as bioblasts, okay, and finally it was Mrs. Carl Blunda. She was the one who actually named it as mitochondria, and till date we are using the very same name. And you know, it has been a while we have been understanding about mitochondria, and I hope you all aware that mitochondria is actually the powerhouse of the cell. Okay. Now, I have a question to everyone, children. Do you know where we will find the respiration process occurring in a cell? Okay, as you are dropping in correctly, of course, it is respiration happens in the mitochondria. Fine. Now, I have a question. Till around 7th or 8th grade, you all have been learning that, you know, digestion process, and it is because of the food you and me get a lot of energy for our activities. Is that true? Okay. So, when you say it is false, so how it is false? Can you get me some reason for that? Okay, I see here some of the responses. Okay, so here it is not actually just by consuming food or by consuming oxygen we get energy. There is a process to be occurred, and that results in the production of energy. One such process is respiration. Okay, it might be confusing to you. Okay, so respiration process. Now, some of you might be wondering how this is helping us to get energy. Well, let me give you an understanding of it. So, respiration is a process which gives us energy. Okay, that's what I am saying. Okay. Now, how it happens, let us see here. So, what happens exactly in respiration is we use oxygen. All of you are aware of that, respiratory gases what we use are basically oxygen and carbon dioxide. So, in the process of respiration we need oxygen, okay. Ultimately this oxygen is combined with a known compound, that is glucose, and that is how we get energy. So, that is the equation, oxygen plus glucose, that is simply how we get energy, okay. So, this is what happens in respiration. Now, it is not only the food. So, we consume food and using that we get to have the glucose. We breathe in, breathing process. When we breathe in we get oxygen and they are to be combined together, and that combination of oxygen and glucose that happens by this particular process, okay. Now, this ultimately happens in this particular organelle of the cell. That is the reason why when this particular process occurs in this particular organelle of the cell, it generates energy, and because it is generating an energy, the form in which it generates energy is actually ATP, which stands for adenosine triphosphate. Adenosine triphosphate, that is the energy currency of the cell, and that is generated by this particular organelle. So, that is why I was telling you we find this organelle in aerobic eukaryotic cells only. Why aerobic? Because they use oxygen. Eukaryotic means the higher order cells, okay. So, that is how we get to have energy using this particular organelle in each and every cell of our body. Now, you see it is actually a rod shaped and double membraned organelle. Outer membrane, because I said double membrane, so it has two membranes, okay. So, one membrane which is outside is having smooth and small pores on its surface, okay, and the inner membrane has finger like projections known as cristae, okay. And the mitochondria has its own DNA which is known as mitochondrial DNA. So, the DNA which it has is of its own type, and that is known as mitochondrial DNA, okay. Now, what you can understand here is, so you have to think here. So, this mitochondrial DNA is something special. Millions of years ago, this mitochondria was actually a separate organism. Later it just combined with the previous heterotrophic cells, and later the game was played differently. So, I would like to tell you that in the upcoming sessions. Now, that is how it happened. That is the reason why they have their own specialized mitochondrial DNA. So, why do we need this? Like I mentioned, it performs cellular respiration to release energy. Because it is releasing energy by this process, so it is known as powerhouse of the cell or powerhouses of the cell, because there are many mitochondria we see in the cell, and it synthesizes some of the enzymes called respiratory enzymes. You know what are enzymes? Can you answer me that? Okay. So, when you see about enzymes, enzymes are those substances, okay, they are kind of catalysts, or in other words actually they are biocatalysts which involve in the respiratory reactions by without undergoing any change in themselves and making the reaction fast, make the reaction occurs in a quite fast pace. So, that helps them to release energy in quite quickly. So, they make these kinds of enzymes. Apart from that, mitochondria is also having a big role in storing calcium, and wherever there is a need within the cell, mitochondria releases the calcium concentration and it sends it to the respective place where it is required. Now, this is how it looks like. This is what is mitochondria you are looking at. So, like I said, it is having its own DNA, okay, and if you see, there are two membranes. So, one is outer membrane, it is the one having the openings, the pores on its surface, and it is quite smooth compared to the inner membrane. So, inner membrane and the outer membrane, there lies a space between them, okay. That space is known as intermembrane space. So, if you can see here, that is a space between the inner membrane and the outer membrane. So, something like this. So, the space between, consider this hand being the outer membrane and this one being the left hand being the inner membrane, there lies a space between these two is actually known as intermembrane space, okay. That is how it is. And you see the inner membrane is actually forming some of the foldings. I am referring to these foldings. Okay, let me use some other color. Okay, this would do good. Okay, these foldings, okay, that is what I am referring here. These foldings are actually known as cristae, and you find the junction there, junction between the two cristae. This is what the junction here referred here, okay. So, these are cristae, and in the mitochondria you will find their own ribosomes, and there is a matrix. Now, this matrix is like a fluid like substance, okay, and in which you will find the granules or ATP synthase, that is an enzyme, ribosomes or DNA, all of those things would be suspended in the matrix. Now, this is for our understanding. So, hope you understood the structure of the mitochondria and the functions and the structure in detail further. So, let us move forward to another organelle, that is endoplasmic reticulum. Now, what do we do with this? So, this endoplasmic reticulum is again an interesting organelle. Just like mitochondria, here also you will find the cristae, cisternae, okay, and it is having vesicles and tubules. So, it has cisternae, I will show you what are they, and you will find the vesicles, okay, and tubules. All this is because of a big network which is formed here in this cell, that too because of the membrane. So, it is also actively known as endomembrane system. So, endoplasmic reticulum, it is actually a big system within the cell which performs several functions. So, this is endomembrane system, okay. So, it has a fluid within itself just like mitochondria we just saw. So, it is endoplasmic reticulum matrix, E matrix or endoplasmic matrix, okay. That is the one which fills the space, and if you would have observed, I have shown you in the structure of the animal cell and plant cell, the endoplasmic reticulum was of different spots, one was with the spots, one was without the spots. Those spots, if there you see those spots there, and that is what we know it as rough endoplasmic reticulum. And the other endoplasmic reticulum structure where you did not find any spots on the surface was actually smooth endoplasmic reticulum. Now, why do we need this? So, endoplasmic reticulum is mainly involved in synthesizing, folding, modification and transportation of proteins. you must have understood. There is on its surface you find the presence of ribosomes, that is why it is known as rough endoplasmic reticulum. And when you see here, it actually synthesizes proteins, it folds the proteins in a required way, it modifies the proteins in a required way, as well as it transports the proteins in a required direction to a specific destination. Okay. So, rough endoplasmic reticulum, as I said, because ribosomes lie on its surface, so hence its RER, and smooth endoplasmic reticulum because it does not have ribosomes on its surface, so it is SER. So, that is quite simple and straight. Hope you have no doubts and confusion. Okay, I see some of your responses, great. So, in case if you have any doubts or something, you can drop in the chat box, I would like to address that. So, like I mentioned, it forms an endomembrane system. That system is actually for intracellular transportation. What do you mean by intracellular transportation? What can you make of it? So, that intracellular transportation, what you see is actually to, like I mentioned, synthesizes protein, folds the protein, modifies the protein and transports the protein. It is for that reason we need it. So, rough endoplasmic reticulum, as you can see, ribosomes are attached on its surface. So, it is rough endoplasmic reticulum. That is the one which actually helps in protein synthesis and transportation. So, most of the part is quite rough and you see doing all these functions. And the one which is smooth endoplasmic reticulum, it involves in synthesizing lipids which are quite closely related to fats. So, they are the ones which are highly related to fats, lipids, okay. And also it helps in removing the poison, or instead of removing the poison, it will also help in neutralizing the poisons. How do you and me take poisons? What can you make about it? Okay, I see some of you are saying like we do not take any poison, but of course, part of the body, there are so many reactions occurring in the body, okay, millions of reactions occur in the body, okay. When in the body you see there are millions of reactions happening, they generate some of the products which are not required by the body. When those products are not required by the body, why do you think they should be present in the body? So, more and more of those products are getting accumulated in the body, that would lead to a problem. All those stuff would turn into actually poisons. So, that is one such way how we are making poisons within the body, because that is a part of the body to make such products and remove them from the body. So, in that case this is helpful for us, detoxification process, okay. And when it comes to, when you are ill, we take medicines, okay. Doctor prescribes medicine for 2 weeks, and what we usually do is, just for taking for 1 week and you stop taking your medication, isn't it? That is what we usually do, most of the people do actually. Now, in that case, if the fever was supposed to exist for 2 weeks, but for first week your fever was gone and you are fine, but still the doctor have advised you to take your medicines for 1 more week. So, it is recommended to consume those medicines for 1 more week. In that course of your medication, you see whatever you are consuming, they are by then may not be required by the body. Then they are nothing but, okay, poisons, because by then your infection might have subsided, we do not need them. So, when these kind of situations comes and this smooth endoplasmic reticulum, it actually detoxifies the lipids. So, that is how it is. So, this is the structure of the endoplasmic reticulum. So, you see there are so many spots on its surface. I would like to show you here. I am referring to these spots encircled in white color, you can see here. So, all this area is having some small spots, they are nothing but the ribosomes, okay. And here you see there are no spots on the surface, okay. So, when you see no spots on the surface, it's quite evident that this is smooth endoplasmic reticulum and this one is having spots because of the presence of ribosome. So, that is rough endoplasmic reticulum. And one point is worthwhile to mention, this endoplasmic reticulum, as I said, it is an endomembrane system, it is having a direct contact with the nucleus, especially with the nuclear envelope. You see, that is the link. Okay. So, what we are trying to do is we are trying to understand the organelles through a sequence. We started with nucleus, we just wanted to learn mitochondria for good reason. Now you see there is a connection between this. So, we also had an understanding of ribosomes in brief, but now we are going to have the very same understanding in detail. Here are we. So, ribosomes are actually the ribonucleoproteins. Okay. So, what do they contain? Ribonucleoprotein particles. Okay. And they were first observed by George Emil Palade. He was a scientist who observed them for the first time. Now, I have a question, but we are trying to understand ribosomes and they are, along with the help of endoplasmic reticulum, they form proteins, everything is fine. But the doubt is, where do we get to see these ribosomes forming in the cell? Or in other words, which component of the cell is known as ribosomal factories? Any guesses? We have already dealt it. Okay, most of you have given correct answer. So, that is nucleoli. So, we just learnt about it. There are several nucleoli present inside the nucleus, and that is where you will see the formation of these ribosome particles. Now, ribosomes are ribonucleoprotein particles, first observed by the George Emil Palade. Okay. It was by Robert B. Roberts in late 1915s. He coined the word ribosome. Okay. So, what are they? They are spherical bodies with two subunits. So, when they have two subunits, okay, they are having a smaller subunit and a larger subunit. And I have to mention one important thing about this. You will not find ribosomes covered by any membrane. There are one such organelles in the plant cell as well as in the animal cells having no membrane covering them. They are just attached in a unit, because you see there are two units present here, the larger subunit and the smaller subunit. So, small subunit and larger subunit here. So, for different cells it is of different types, starting here prokaryotic cell and the eukaryotic cell. You see that prokaryotic cell is of having the smaller subunit with a unit 30S and the larger subunit in the prokaryotic cell is of 50S, collectively known as, this type of ribosome is 70S ribosome. Okay, got my point? Now, next you see eukaryotic cell, the smaller unit is 40S, larger unit is 60S. Now, as a whole, the eukaryotic cell's ribosome is having a type of 80S type. Yeah, I see some of the questions arising here. What is that S standing next to these numbers? And that is what we are here to understand. So, I will show you what are they, but before you have to look at this. So, this is what is ribosome would look like, okay, and you are not finding any type of membrane covering these. They are just like this, okay. So, as the size indicates you, this is the larger subunit and this is the smaller subunit. Same stands for this particular ribosome also, okay. So, this one belongs to prokaryotic ribosome, which is 70S type, and this one is a eukaryotic ribosome having the 80S type. Now, do not break your head on that S. I would like to show you what is that S here actually. So, if you have a look here, S stands for the unit called Swedberg unit. okay. It is denoted by S. In some books you see, in some publications you see it is denoted by S, and in some publications you see it is mentioned as S followed by lowercase v. It is a non-SI metric unit for sedimentation coefficients. Now, can anyone just get me what do you mean by sedimentation coefficient? Okay, so okay, I get it. Some of you are dropping in the answers. Okay, sedimentation coefficient. Let me tell you something here. So, Swedberg's unit, like I mentioned, it offers a measure of a particular size. Sorry, the size of a particle. Now, when it comes to these, they are very small particles. How small I can show you here, because previously I shown you the structure of the endoplasmic reticulum. I have told you that there are small dot like structures on the surface. So, this itself can make you understand this is a big, you know, it is a membranous structure inside the cell. On the surface of this structure you see the presence of ribosomes. So, that itself is quite clear to us that they are really very small. Ribosomes are very small, and you see them occupying this particular place on the endoplasmic reticulum. Okay. So, when that is the case, based on this particle's size, that is the ribosome's size, you cannot actually measure its, you know, diameter or the, you know, length or breadth or something like radius or something. It is not possible. Now, what we will be doing is we will try to find its size by an indirect method. You know, there is a process called centrifugation. I don't know how many of you are aware of it. I am writing on the screen. It is done in a centrifuge. It's a okay. There are different types of centrifuges. Sometimes electrical, mechanical. So there are electronics. So there are so many types of there. Okay. So centrifugation is a process. If I have to make you understand kind of centrifugation, you know, when I was a child, okay, whenever, you know, we used to wash our clothes, we used to put the, you know, obviously we drop in the water here and we put the detergent, the washing powder, right, detergent. When we do that, we try to swirl the water, okay, using your hand you try to create a whirlpool like thing inside the water, isn't it? So, in that bucket, that is what we do it. Now, what you observe by doing that, you see that and it forms you know, a whirlpool like arrangement, and the water is moving away. Okay. Now, that is a bigger scenario, but here what happens in centrifugation, some test tubes are used, okay, different test tubes are used, and in that, you know, the required solution is dropped in with the particles like this, and they will see, they will, you know, rotate it with a different speed, RPM, rotations per minute or revolutions per minute, okay, and they will see that they will make sure the particles are disturbed heavily, and after that they will wait for some time to settle down the particles which were here because of the rotation. Once if they get, you know, settled somewhere here, they will record at time t1 and then at time t2 what is the difference. They will try to find those kind of differences using that, you know, after rotating this for quite a long time in the centrifuge, and you will see it takes time to settle down from particle from here to here, that is time t1 to t2. Based on that, indirectly it gives an understanding that, you know, larger the size of the particle, it takes bit quicker time to settle down. Small the size of the particle, it takes more time to settle down. Now, that is what happens here. Using that technology, that is centrifugation, okay, here you see indirectly based on its sedimentation rate, that means how quickly this particular particle under some kind of acceleration it is settling down at the bottom. Okay? You see how fast a particle of given size and shape settles to the bottom of a solution. This is how the size of these particles would be measured, because you and me cannot see them directly, so that we can measure their grain size using some scales or something. So, because they are very, very small, they are so small that you see them having attached to this particular structure. That is the reason why you see them, we use a unit called Swedberg's unit, which is a non-SI metric unit. So, hope you got my point today's discussion, and why they are having no membrane on their surface. Now, why they are having no membrane on their surface is a simple reason, because they have to run this particular system through this path. So, that is the reason why they are having no membrane on them, and using this they try to make protein. So, that is what you see, there is a growing protein over here. Okay. By the way, mRNA stands for messenger RNA, tRNA stands for transfer RNA. Okay. So, that is the reason why they are having no membrane. So, hope you understood this session. Now, what we will be doing is, we will try to look at the content whatever we have discussed so far. So, in this particular session, we try to understand the constituents of the plant cell and an animal cell. Using that, we try to differentiate those striking features of the plant cell and animal cell, and then we had an elaboration on the powerhouse of the cell, which is nothing but mitochondria. Mitochondria are known as the powerhouses of the cell, and then we explored the detailed structure of endoplasmic reticulum, and we understood its function, what does it doing for the cell. So, if you remember, we saw them, they were of different types, RER and SER. RER is a rough endoplasmic reticulum, SER is smooth endoplasmic reticulum. Okay. Finally, we just saw the structure and functions of ribosomes. So, this is what we discussed in this particular session. In the upcoming sessions, we will be having an understanding of some more organelles. So, try to understand, you can get me the doubts if you have any, and we will discuss those doubts in the upcoming sessions. Thank you so much.